Hello there, and welcome to the Slumber Science Society. Tonight, we're going on a journey through deep time to an age of giants. An age when the world was gripped by ice, when mammoths roamed the frozen plains, and when saber-toothed cats prowled through twilight forests. This is the story of discovery, of the scientists and explorers who unearthed these ancient creatures, and of the remarkable animals themselves, frozen in time, waiting to tell their stories. So get comfortable, settle in, and let's begin our exploration of the Ice Age animals that once walked this earth. In the frozen heart of Siberia, where permafrost grips the land for most of the year, something extraordinary happens when the brief summer sun thaws the surface. Ancient secrets emerge from the ice, preserved for tens of thousands of years. It was here, in these remote northern reaches, that humans first began to understand that the world was once home to creatures unlike anything alive today. The native peoples of Siberia had long known about the bones that emerged from riverbanks and eroded cliffs. They developed their own explanations for these mysterious remains. Some believed that the enormous curved tusks they found were actually the claws of giant birds that burrowed underground. Others thought they were the remains of enormous rats that lived beneath the earth and died when they emerged into sunlight. These weren't foolish superstitions, they were reasonable attempts to explain the inexplicable, to make sense of bones that belonged to no living animal they knew. But it wasn't just in Siberia where these discoveries sparked wonder and confusion. In 1335, in the Austrian city of Klagenfurt, workers found an enormous skull in a nearby quarry. The skull was massive, with a strange horn-like protrusion, and the people of the time were certain they had discovered the remains of a dragon. This wasn't fantasy to them, it was evidence. Dragons, after all, featured prominently in their mythology and religious texts. What else could such a skull belong to? In 1590, the city commissioned a statue of a lindworm, a dragon-like creature from local legend, and the sculptor used this ancient skull as his model for the head. The statue still stands in Klagenfurt today, a limestone dragon with a head based on a woolly rhinoceros that died more than 30,000 years ago. These early encounters with Ice Age remains shaped human understanding in profound ways. They seem to confirm ancient stories of monsters and giants. They suggested that the world had once been a very different place, populated by creatures of immense size and power. But the true nature of these remains, and the story they told about Earth's past, would take centuries more to understand. The scientific investigation of these frozen giants truly began in the 18th century, when European naturalists started to travel to Siberia specifically to study these mysterious remains. The Russian Empire, which controlled these vast territories, encouraged such expeditions as part of broader efforts to catalogue and understand their enormous domain. One of the most important of these early explorers was Peter Simon Pallas, a German naturalist who traveled extensively through Siberia between 1768 and 1774. Pallas was a meticulous observer and a careful scientist, and he brought a new level of rigor to the study of these ancient bones. In 1769, during his Siberian expeditions, he wrote the first scientific description of a woolly rhinoceros, based on specimens he had observed. But Pallas didn't just describe what he saw in the field. He actively collected specimens to send back to museums and scientific institutions where they could be properly studied. In 1772, while in the region around Irkutsk in eastern Siberia, he acquired something remarkable from local inhabitants. They had discovered the head and two legs of a woolly rhinoceros, preserved in the permafrost. Pallas named this specimen Rhinoceros lenonensis, after the Lena River near where it was found. This wasn't just a collection of bones, the preservation was extraordinary, with soft tissue still attached. The specimen Pallas acquired had been found in 1771 in the region near the Viliui River, a tributary of the Lena. Along with the head and legs, there were pieces of hide still covered with the animal's thick fur. These remains were carefully packaged and sent to the Kunstkamera in St. Petersburg, where they became one of the founding specimens of what would eventually become one of the world's great natural history collections. For the first time, European scientists could study not just the skeleton of an extinct animal, but its actual flesh and fur. This discovery was revolutionary. The preservation was so complete that scientists could see what this animal had actually looked like in life. They could examine the texture of its skin, the nature of its fur, the structure of its feet. The woolly rhinoceros wasn't just a theoretical reconstruction based on bones, it was real, tangible, and remarkably well preserved. The animal had a thick coat of brown fur, shaggy and dense, clearly adapted for life in frigid conditions. Its feet were broad and sturdy, designed to carry a massive weight across snow and ice. And most striking of all were its horns, not growing from bone like the horns of cattle, but made of compacted keratin, the same material as human fingernails, growing from the skin of its snout. Other specimens continued to emerge from the Siberian permafrost throughout the late 18th and early 19th centuries. In 1877, another head and leg were discovered, this time from a tributary of the Yona River, further east in Siberia. Each new discovery added to the growing understanding of these vanished animals and the world they inhabited. But it was Johann Friedrich Blumenbach, a German physician and naturalist, who gave the woolly rhinoceros its scientific name in 1799. He called it Rhinoceros antiquitatis, the ancient rhinoceros. The name captured both its relationship to modern rhinoceroses and its extreme age. Blumenbach was one of the founders of comparative anatomy, and he recognized that this extinct creature, while clearly related to living rhinoceroses, was also distinctly different, adapted for a very different world. The scientific classification of the woolly rhinoceros was refined further in 1831 when Heinrich Georg Bronn, studying the animal's teeth, recognized that the differences from modern rhinoceroses were substantial enough to warrant placing it in its own genus. He moved the species to the genus Celodonta, where it remains today. The full scientific name became Celodonta antiquitatis, and this Ice Age giant took its place in the growing catalogue of extinct megafauna that scientists were slowly reconstructing. These weren't isolated finds. Throughout the 19th century, the rivers and streams of Siberia continued to yield their frozen secrets. Every spring thaw, every erosional event, every landslide had the potential to reveal another piece of the puzzle. Local inhabitants became adept at recognizing these remains and understood that scientists would pay good money for well-preserved specimens. A network developed where locals would collect bones, tusks, and occasionally more complete remains, which would then make their way through traders to major cities and eventually to museums and universities. The animals emerging from the Siberian ice painted a picture of a world very different from the one we know. This wasn't a warm, tropical past. These animals were adapted for cold, for ice, for the harsh conditions of glacial periods. Their thick fur, their compact bodies designed to conserve heat, their specialized features for surviving in frozen landscapes, all of this told a story that challenged the understanding of Earth's history. Scientists began to realize that the planet's climate had not been constant. There had been periods when ice covered much of the northern continents, when the sea level was lower because so much water was locked up in glacier, when the landscape and ecology were fundamentally different from today. The animals preserved in Siberian permafrost were witnesses to these vanished ice ages, and their remains were the key to understanding these ancient climates. The woolly rhinoceros that Pallas had first described scientifically turned out to be just one of many species that had thrived during the Ice Age. But it was a particularly important one, both because of the quality of preservation in Siberia and because of how widespread it had been. This was not just a Siberian animal. Woolly rhinoceros remains were found across Europe as well, from Britain to the Mediterranean, from Spain to the Caucasus. During the coldest periods of the Ice Age, roughly between 450,000 years ago and 14,000 years ago, these creatures were one of the most successful large mammals in the Northern Hemisphere. The woolly rhinoceros was an impressive animal by any measure. From head to tail, it measured between 3.2 and 3.6 meters, roughly 10 to 12 feet. At the shoulder, it stood between 1.45 and 1.6 meters tall, about 5 feet, comparable to a modern white rhinoceros. But what really set it apart were its proportions and specialized features. The animal was massively built, weighing between 1.5 and 2 metric tons, with some estimates suggesting the largest individuals may have reached as much as 2.9 tons. But size wasn't the woolly rhinoceros's most distinctive feature. That honor belonged to its horns. The animal had 2 horns, both made of compacted keratin. The front horn, growing from the nasal bone, was exceptionally long. Typical specimens had front horns measuring between 1 meter and 1.35 meters, over 4 feet long. But the longest front horn ever recorded measured an astonishing 164.7 centimeters, nearly 5.5 feet. This wasn't a horn designed for combat alone, though it certainly could be used that way. The horn was often flattened in cross-section, particularly near the tip, and evidence suggests the animal used it to brush away snow when grazing, sweeping aside the winter covering to reach the grasses underneath. The second horn, growing from the frontal bone further back on the skull, was smaller, typically reaching up to 4 to 7.5 centimeters in length. Together, these 2 horns gave the woolly rhinoceros a distinctive profile, one that was captured in remarkable detail in Ice Age cave paintings, as we'll explore later. The skull of the woolly rhinoceros was massive, measuring 70 to 90 centimeters in length, with an enormous lower jaw that could reach 60 centimeters. But what made the skull truly unique among rhinoceroses was its ossified nasal septum. In most mammals, the septum dividing the 2 nostrils is made of cartilage, but in the woolly rhinoceros, this septum had turned to bone, creating an incredibly strong structure that could support the weight of that enormous front horn and withstand the stresses of using it to sweep snow. The teeth of the woolly rhinoceros were highly specialized as well. The animal had high-crowned molars with very thick enamel, perfect for grinding the tough, abrasive grasses that formed the bulk of its diet. Interestingly, adults lacked incisors entirely. The front of the mouth was simply a broad, tough pad that worked with the lower jaw to crop vegetation. This is similar to modern grazing animals like cattle and horses, and it tells us something important about the woolly rhinoceros's feeding strategy. The animal was covered in a thick coat of long, shaggy brown fur in adults, while juveniles had lighter brown coats. This wasn't just a light covering, this was serious cold weather insulation. The outer layer of long guard hairs protected against wind and moisture, while beneath was a dense undercoat that trapped air and provided exceptional insulation. On its back, the woolly rhinoceros had a pronounced hump, similar to what we see in bison. This hump was composed of fat and served multiple purposes. It was an energy reserve for lean times, and it also played a role in thermoregulation, helping the animal maintain its body temperature in the extreme cold of Ice Age winters. But what was the world like that the woolly rhinoceros inhabited? This is where our understanding of Ice Age ecology becomes crucial. The woolly rhinoceros was a creature of what scientists call the mammoth steppe, a vast ecosystem that stretched from Western Europe clear across Northern Asia and into North America during glacial periods. This was a landscape unlike anything that exists today, a cold, dry grassland that supported enormous herds of large herbivores. The mammoth steppe wasn't the frozen wasteland that the term Ice Age might suggest. It was a productive ecosystem, though a harsh one. The grasses and sedges that carpeted this landscape were highly nutritious, and they could support large populations of grazing animals. The woolly rhinoceros shared this landscape with woolly mammoths, of course, but also with steppe bison, reindeer, saiga antelope, and the magnificent Irish elk with its enormous palmate antlers. Scientists sometimes refer to this assemblage as the Mammothus-Celadonta faunal complex, named after the scientific names for the woolly mammoth and woolly rhinoceros, the two most characteristic species. The woolly rhinoceros was primarily a grazer, using its wide upper lip, similar to that of the modern white rhinoceros, to crop grasses and sedges efficiently. But it wasn't exclusively a grass eater. Analysis of stomach contents from frozen specimens has revealed that the animal also consumed woody plants, including conifers, willows, and alders, along with flowers, forbs, and even mosses. This dietary flexibility was probably important for survival in an environment where food availability changed dramatically with the seasons. The animal preferred lowlands, plateaus, and river valleys, generally avoiding mountainous terrain. It thrived in areas with dry to arid climates during the glacial periods, the kind of environment that supported the mammoth steppe ecosystem. Life history studies based on growth rings in the horn keratin suggest that woolly rhinoceroses could live for 40 years or more, a considerable lifespan for such a large mammal. Growth patterns show that young woolly rhinoceroses grew most intensively during their first 3 to 4 years, reaching young adulthood at 7 to 10 years, and achieving full maturity at 14 years or older. Females had 2 teats, suggesting they typically raised 1 calf at a time, though twins may have occurred occasionally. The long interbirth interval, probably 2 to 3 years, meant that populations grew slowly and were vulnerable to disruption. Despite their massive size, young woolly rhinoceroses were vulnerable to predation. Cave hyenas and cave lions, both considerably larger than their modern relatives, preyed on calves and juveniles. But adult woolly rhinoceroses had few natural predators. The combination of size, aggressive temperament, and those formidable horns made them a dangerous target for any predator. The horns served multiple purposes beyond defense. Males likely used them in combat with other males over territory or mates, similar to how modern rhinoceroses use their horns. The wear patterns on some fossil horns suggest vigorous use, with grooves and scratches that indicate repeated impact. Bulls were probably territorial, like most modern rhinoceroses, defending areas of good grazing from other males while allowing females to pass through. But the question that fascinates us most about these remarkable animals is not just how they lived, but when and why they disappeared. The woolly rhinoceros was remarkably successful for hundreds of thousands of years, thriving across the vast expanse of northern Eurasia through multiple glacial and interglacial cycles. But around 14,000 years ago, they vanished. The extinction of the woolly rhinoceros wasn't sudden everywhere. Analysis of nuclear genomes from multiple specimens shows that the population actually began expanding around 30,000 years ago, during a period when many other megafauna species were already declining. This suggests that the woolly rhinoceros was well adapted to Ice Age conditions and was actually thriving as recently as 30,000 years ago. But then, between 17,000 and 15,000 years ago, something changed. The woolly rhinoceros began to disappear from its range, starting in Western Europe and moving eastward. By 15,000 years ago, the species was gone from Europe entirely. The last reliable records from the Ural Mountains date to about 14,200 years ago, while the youngest remains from northeastern Siberia are approximately 14,000 years old. This timing is significant because it coincides with a major warming event called the Bolling-Allerod interstadial, a period of rapid warming that disrupted the mammoth steppe ecosystem. As temperatures rose, the dry grasslands that woolly rhinoceroses depended on began to shrink and fragment. Forests expanded into former grazing grounds. The deep snows that came with warmer, wetter conditions were particularly problematic. Woolly rhinoceroses, despite their adaptations for cold, struggled to move through deep snow and couldn't efficiently forage in forested environments. Genetic analysis of some of the last woolly rhinoceroses shows that populations maintained substantial genetic diversity right up until near the end, suggesting that they weren't declining slowly over many generations but rather faced relatively rapid habitat loss and fragmentation. Some North Sea specimens show high rates of cervical rib abnormalities, which might indicate inbreeding or environmental stress in these late populations as suitable habitat became scarce. Interestingly, there's controversial evidence from environmental DNA studies suggesting that woolly rhinoceroses might have persisted in northeastern Siberia until as recently as 9,800 years ago, well into the Holocene. But this evidence is disputed because ancient DNA can be mobile in permafrost, potentially contaminating younger sediments, so most scientists consider the 14,000-year date to be more reliable for the species' extinction. The role of humans in the woolly rhinoceros's extinction is debated. Unlike mammoths, woolly rhinoceros remains are relatively rare at human archaeological sites. Only about 11% of Siberian prehistoric sites contain rhinoceros remains or images. There is clear evidence that both Neanderthals during the Middle Paleolithic and modern humans during the Upper Paleolithic hunted woolly rhinoceroses. Bones have been found with weapon marks, and in one case, a spear was found directly associated with a carcass. At Gudenus Cave in Austria and Königsau in Germany, archaeologists have found woolly rhinoceros bones that were heavily beaten and show slash marks, evidence that humans were extracting bone marrow. Horns and bones were used to make tools and weapons. At a site called Zwolen in Poland, a device was fashioned from a battered pelvis bone. Most dramatically, at the Yona rhinoceros horn site in Siberia, archaeologists discovered 27,000-year-old spear throwers carved from woolly rhinoceros horn, some of the most sophisticated tools of the Upper Paleolithic. But the most remarkable human artifact made from woolly rhinoceros remains comes from Bolshoi-Lyakovsky Island in the far north of Siberia. There, archaeologists discovered a spear with a tip made from woolly rhinoceros horn, dated to 13,300 years ago. This is the furthest north human artifact ever found, evidence that people were hunting these animals in some of the most extreme environments on Earth right up until the species' final days. Yet despite this evidence of hunting, most scientists believe that climate change and habitat loss, not human predation, were the primary drivers of woolly rhinoceros extinction. The animals had coexisted with human hunters for tens of thousands of years during earlier cold periods. What changed wasn't the intensity of hunting, but the ecosystem itself. As the mammoth steppe fragmented and disappeared, so too did the woolly rhinoceros. But the woolly rhinoceros wasn't alone in the Ice Age bestiary. Far to the west, in what is now Southern California, a very different story of discovery was unfolding, one that would reveal an ecosystem even richer and more diverse than the Siberian mammoth steppe. In 1901, a geologist named William Warren Orcutt was exploring an area of Los Angeles called Rancho La Brea. The land was marked by pools and seeps of thick, black asphalt that bubbled up from the ground, creating sticky, treacherous traps. Locals had long known about these asphalt deposits. In fact, the name La Brea literally means the tar in Spanish, so Rancho La Brea is a bit redundant. It essentially means the ranch of the tar. Orcutt, however, noticed something that others had overlooked. Embedded in the asphalt were bones, lots of bones, and they weren't modern. These were the fossilized remains of animals from the deep past, perfectly preserved by the asphalt that had trapped them thousands of years ago. The discovery at La Brea would transform our understanding of Ice Age North America. But it wasn't Orcutt who conducted the first systematic excavations. That honor belongs to John Campbell Merriam, a paleontologist from the University of California, Berkeley. In 1913, Merriam began what would become one of the most extensive and productive paleontological excavations in history. What Merriam and his team discovered over the following years was extraordinary. In just the first 2 years of systematic excavation, they recovered 750,000 specimens from 96 separate sites within Rancho La Brea. 750,000 specimens. Think about that number for a moment. This wasn't just a fossil site, it was a complete window into an entire ecosystem, preserved in exquisite detail. But what made La Brea truly unique was the nature of what was preserved there. At most fossil sites, herbivores vastly outnumber carnivores, which makes sense. In any ecosystem, there are always more prey animals than predators. But at La Brea, the situation was dramatically different. Approximately 90% of the excavated bones belonged to predators. This unusual ratio tells us something important about how the site formed. The asphalt seeps at Rancho La Brea were natural traps. During the Pleistocene, when the area was more wooded and had a cooler, wetter climate than modern Los Angeles, large herbivores would occasionally become mired in these sticky pools. Unable to escape, they would die, their bodies sinking into the asphalt. But their death cries and the scent of their remains would attract predators and scavengers from miles around. The saber-toothed cats, the dire wolves, the short-faced bears, all of them would be drawn to what seemed like an easy meal. They would venture onto the asphalt-covered ground, perhaps not recognizing the danger until it was too late. Their weight would break through any surface layer hardened by the sun, and they too would become trapped. More death, more scent, more predators attracted, more deaths. The asphalt seeps at La Brea operated as predator traps for tens of thousands of years, accumulating an extraordinary concentration of carnivore fossils. The fossils at La Brea span a considerable time period. Radiocarbon dating shows that the oldest materials are approximately 38,000 years old, but the majority of the fossils date to between 20,000 and 10,000 years ago, with peak accumulation during the height of the last glacial maximum. But what exactly was being preserved in such abundance at La Brea? The most common fossil is that of the dire wolf, Inocyne-dirus. These were not the gray wolves we know today. They were larger, more robust, with more powerful jaws and teeth adapted for crushing bone. More than 166,000 dire wolf specimens have been recovered from La Brea. 166,000. That's more dire wolf fossils than exist for any other species from any site anywhere in the world. But La Brea is perhaps most famous for a different predator, the saber-toothed cat. When most people think of Ice Age carnivores, they picture the saber-tooth, and with good reason. The fossils from La Brea represent the species Smilodon fatalis, one of 3 saber-toothed cat species in the genus Smilodon. And La Brea has yielded more saber-toothed cat fossils than all other sites in the world combined. More than 166,000 Smilodon fatalis specimens have been recovered from the Rancho La Brea tar pits, a collection so vast that it has allowed scientists to study not just the anatomy of these animals, but their population biology, their pathology, their growth rates, and even their social behavior. Smilodon fatalis was a formidable predator by any measure. These cats lived in North America from roughly 1.6 million years ago until about 10,000 years ago. An adult Smilodon fatalis weighed between 160 and 280 kilograms, roughly 350 to 600 pounds, making them comparable in weight to a modern African lion, though much more robustly built. At the shoulder, Smilodon fatalis stood about 100 centimeters, just over 3 feet, but this doesn't capture how powerful these animals were. They had exceptionally well-developed forelimbs, much more robust than any modern cat, with massive shoulder and arm muscles. The scapula, or shoulder blade, was positioned high on the back, creating attachment points for enormous muscles that could pull downward with tremendous force. But of course, the most distinctive feature of Smilodon was its teeth. The upper canines were elongated into blade-like sabers that could reach up to 18 centimeters in length, 7 inches of dagger protruding from the upper jaw. In the largest species of Smilodon, Smilodon populator from South America, these canines could reach an incredible 28 centimeters, nearly 11 inches. These weren't stabbing weapons in the way we might first imagine. The canines were relatively thin and fragile, despite their length. They were precision instruments, not brute force tools. The jaw structure of Smilodon was specialized to protect these teeth while using them effectively. The cat could open its jaws to an angle of more than 110 degrees, compared to about 65 degrees in modern lions. This extreme gape allowed the saber teeth to clear the lower jaw and be driven into prey without risking breakage. The killing technique of Smilodon was quite different from modern big cats. A lion or tiger kills by delivering a suffocating bite to the throat, using their powerful jaws to clamp down and hold until the prey stops breathing. Smilodon couldn't do this. Its bite force was actually weaker than that of modern big cats. Instead, Smilodon used a technique called the canine shear bite. The cat would use its powerful forelimbs to wrestle prey to the ground and hold it immobile. Then, with a flexion of the neck and a rotation of the skull, it would drive those blade-like canines into the throat of its prey, severing the major blood vessels and windpipe. Death would come quickly from blood loss and shock. The robust forelimbs and heavily muscled shoulders were essential for this hunting strategy. Smilodon had to be able to grapple with and control prey that was often much larger than itself. The animal's proportions reflect this. It had a relatively short, compact body with reduced lumbar vertebrae, sacrificing some flexibility for power and stability. The prey of Smilodon fatalis at La Brea is known from multiple lines of evidence. Isotope studies, which look at the chemical signatures in bones and teeth that reflect diet, show that Smilodon primarily hunted large herbivores. The most common prey items were bison, specifically Bison antiquus, the ancestor of modern American bison but considerably larger, and camelops, a large camel that lived in North America during the Pleistocene. Ground sloths, particularly Paramylodon harlani, were also important prey items. Interestingly, the isotope studies also reveal that Smilodon had significant dietary overlap with dire wolves and American lions, both of which were also common predators at La Brea. However, there were subtle differences. Smilodon seemed to prefer prey from more forested areas, including deer and forest-dwelling bison, while dire wolves tended toward prey from more open grasslands. This suggests some degree of ecological separation between these predators, reducing direct competition. One of the most fascinating aspects of the La Brea Smilodon fossils is what they reveal about social behavior. In modern big cats, only lions are truly social, living in prides where related females hunt cooperatively and males defend territories. Tigers, leopards, and other cats are largely solitary, coming together only to mate. For a long time, scientists debated whether Smilodon was social or solitary. The sheer number of individuals at La Brea suggests they might have been social if they responded to the distress calls of trapped pack members, that would explain why so many individuals ended up in the tar pits. But this is indirect evidence. More compelling evidence comes from the fossils themselves. Many Smilodon specimens from La Brea show healed injuries, including serious trauma to limbs and jaws. A solitary predator with a broken limb would likely starve, unable to hunt effectively. But if Smilodon lived in social groups, an injured individual could be provisioned by other group members until it healed. The high frequency of healed injuries in the La Brea population suggests this kind of social support. Even more intriguing is evidence from dental injuries. Some Smilodon specimens show patterns of tooth wear that indicate they were eating softer flesh, avoiding the kind of bone chewing that would normally be expected. This suggests that group members might have been providing choice cuts of meat to injured individuals who couldn't effectively process carcasses themselves. Most dramatically, in 2021, paleontologists described 3 associated Smilodon skeletons from Ecuador, an adult female and 2 subadults, 1 male and 1 female. The subadults were approximately 2 years old and were still with their mother at the time of death. This suggests extended parental care, with young Smilodon remaining dependent on their mother for a considerable period, similar to what we see in social modern cats like lions. The rarity of juvenile Smilodon at La Brea despite the abundance of adults supports this interpretation. If young cats were being kept at dens while adults hunted, they wouldn't be present at kill sites to become trapped in the tar pits. This is exactly the pattern we'd expect for a social species with extended parental care. The growth and development of Smilodon was a lengthy process. Unlike modern big cats, which reach adult size relatively quickly, Smilodon took about 3 years to reach full size. This extended development period was probably necessary because of those remarkable canine teeth. A remarkable study published in 2025 revealed something extraordinary about Smilodon development. Young Smilodon were born with a set of milk canines, smaller versions of the adult sabers. But rather than losing these baby teeth before the adult teeth came in, as most mammals do, young Smilodon retained their milk canines for approximately 30 months, even as the adult teeth were erupting. For a substantial period of the animal's growth, it was double-fanged, with both sets of canines present simultaneously. This wasn't an accident of development, it was an adaptation. The juvenile milk canines helped to reinforce the emerging adult canines, reducing the bending strain on these developing teeth. The adult sabers, remember, were relatively fragile precision instruments. Having the milk teeth present during their eruption and initial growth protected them from damage at their most vulnerable stage. Despite their impressive adaptations, life for Smilodon wasn't easy, and the La Brea fossils tell this story as well. Detailed studies of 1,000 Smilodon skulls from the site revealed that 36% showed erosion of the parietal bones, where the massive jaw muscles attached. This erosion was caused by repeated stress from the constant contraction of these muscles during hunting and feeding. Many specimens show microfractures in the skull from the repeated impacts involved in driving those canine teeth into prey. The bones of the shoulder show distinctive bony growths at the deltoid muscle insertion points, evidence of repeated strain from the forceful pulling down of prey. Sternum injuries are also common, likely from collisions with prey during the initial grappling stage of the hunt. The overall trauma frequency in Smilodon fossils from La Brea is 4.3%, compared to 2.8% in dire wolves from the same site. Smilodon seems to have lived a more physically demanding life, with more frequent injuries than even the pack-hunting dire wolves. Shoulder injuries and trauma to lumbar vertebrae are particularly common, reflecting the intense physical demands of the grappling hunting strategy. As the end of the Ice Age approached, the Smilodon population at La Brea began to show signs of stress. Some late-period specimens show substantially decreased mandibular bone thickness, which might indicate nutritional stress. High rates of subchondral defects in limb joints, possibly indicating inbreeding, appear in the latest fossils. These are the signatures of a population under pressure, dealing with changing environmental conditions and possibly a declining prey base. The youngest reliable Smilodon fatalis fossils from La Brea are dated to 13,025 years ago. Other reliable dates from Iowa place Smilodon there as recently as 13,600 to 13,450 years before present. These dates are significant because they suggest that Smilodon went extinct before the Younger Dryas, a period of intense cold that occurred between 12,900 and 11,700 years ago. In other words, it wasn't a return to glacial conditions that killed off Smilodon, it was the warming that preceded it. The extinction of Smilodon across the Americas is part of the broader pattern of megafauna extinction at the end of the Pleistocene. As large herbivores declined, specialist predators like Smilodon, which had evolved specifically to hunt very large prey, found themselves without adequate food resources. Their specialization, so successful for over 1 million years, became a liability when the ecosystem changed rapidly. But La Brea preserved more than just predators. The site has yielded remains of more than 230 different species, from tiny rodents and birds to giant ground sloths and mammoths. In 1977, the George C. Page Museum opened next to the tar pits, specifically to house and display the incredible wealth of fossils recovered from the site. Today, excavation continues at Pit 91, which has been in continuous operation, uncovering new specimens and new insights into Ice Age California. One of the most remarkable recent discoveries came in 2009 during construction of the museum's parking garage. The excavation revealed a nearly complete mammoth skeleton, nicknamed Zed, along with numerous other fossils. The construction had to be halted while paleontologists carefully excavated the remains, a reminder that even in the heart of modern Los Angeles, the ancient past lies just beneath the surface. There's even a human connection to the La Brea tar pits. Among all the hundreds of thousands of animal bones, only one human skeleton has ever been recovered. Known as the La Brea woman, this individual died approximately 10,000 years ago, one of the last chapters in the story of Pleistocene Los Angeles. How she ended up in the tar pits remains a mystery. Was it an accident? Some form of violence? We may never know, but her presence reminds us that humans were part of this ecosystem, witnessing and interacting with these now extinct animals. The story of La Brea is ultimately about ecosystem collapse. For tens of thousands of years, Southern California supported a diverse assemblage of large predators and the prey that sustained them. But as the climate warmed and changed at the end of the Pleistocene, this ecosystem couldn't survive. The large herbivores declined, and with them went the specialized predators. The asphalt seeps that had trapped so many animals finally fell silent, no longer claiming victims because there were no longer megafauna to trap. While the scientists at La Brea were uncovering the predators of Ice Age California across the Atlantic, in the limestone caves of southern Europe, another window into the Ice Age was opening. This one would reveal not just the animals themselves, but how humans saw them, depicted them, and perhaps understood them. In September of 1940, in the Dordogne region of southwestern France near the village of Montagnac, an 18-year-old man named Marcel Ravedat was exploring the woods with 3 friends. Ravedat's dog had disappeared down a hole, and while investigating, the young men discovered a narrow shaft that opened into the hillside. Armed with a makeshift lamp, they descended into the darkness. What they found took their breath away. The walls of the cave were covered in paintings. Not crude scratchings, but sophisticated, beautiful depictions of animals rendered in ochre, charcoal, and manganese oxide. Horses, aurochs, deer, and more, hundreds of individual images covering the walls and ceiling of multiple chambers. They had discovered Lascaux's cave, one of the most spectacular examples of Upper Paleolithic art ever found. Over the following weeks and months, as archaeologists and prehistorians examined the cave, the full scope of the discovery became clear. The cave contained nearly 6,000 figures, including about 1,900 identifiable animal images. The quality of the art was stunning. In what's now called the Hall of the Bulls, enormous aurochs, the wild ancestors of domestic cattle, parade across the ceiling, some measuring up to 5.2 meters in length. These aren't simple outlines, they are rendered with understanding of anatomy, with a sense of movement and weight, with subtle shading that gives the animals dimension and presence. The paintings aren't recent. Using radiocarbon dating on charcoal from the cave walls and floor, scientists determined that the art was created between 17,000 and 22,000 years ago, during the Upper Paleolithic. The humans who created these images were living in the midst of the last glacial maximum, when ice sheets covered much of Europe and the climate was considerably colder than today. The animals depicted at Lascaux's include horses, aurochs, deer, ibex, and more. But notably, they also include now extinct Ice Age megafauna. The artists who painted Lascaux's lived alongside these animals. They hunted them, feared them, and clearly studied them closely enough to depict them with remarkable accuracy. Lascaux's became one of France's most popular tourist attractions, but the cave was closed to the public in 1963 after it became clear that the breath, body heat, and carbon dioxide from thousands of visitors were damaging the paintings. Today, visitors see perfect replicas, Lascaux's 2 and Lascaux's 4, while the original cave is carefully monitored and preserved for future generations. But Lascaux's, while spectacular, is not unique. All across Europe, from Spain to the Ural Mountains, from France to Italy, caves contain Ice Age art depicting the animals of the Pleistocene. And among these animals, the woolly rhinoceros holds a special place. Before the discovery of Chauvet Cave in southern France, only about 20 Paleolithic drawings of woolly rhinoceros were known from European caves. These drawings showed the animal's distinctive features: the raised back with its fatty hump, the low head position, the curved ears, and most dramatically, the long, curved front horn. Some drawings showed the animal's shaggy coat indicated by careful lines, giving texture to the image. Many paintings showed a black band dividing the body, though whether this represents actual coloration or is stylistic is debated. But in 1994, when Jean-Marie Chauvet and his team discovered the cave that now bears his name, that number increased dramatically. Chauvet Cave contains some of the oldest cave art known, with radiocarbon dates of over 31,000 years for some panels. And among the hundreds of animals depicted in Chauvet, woolly rhinoceroses feature prominently. One of the most famous images from Chauvet shows 2 woolly rhinoceroses in what appears to be combat, their horns locked as they push against each other. This isn't speculation about behavior, it's direct observation by people who had seen such fights. The detail is extraordinary. You can see the hump on the back, the specific shape of the horn, the position of the ears. These artists knew woolly rhinoceroses intimately. Cave art depicting woolly rhinoceros has been found at Rouffignac, at Lascaux's, at Font-de-Gorme in France, and as far east as Kapova Cave in the Ural Mountains. Some images show woolly rhinoceroses struck with spears or arrows, direct depictions of hunting. But many images show the animals simply being animals, walking, grazing, or interacting with each other. But cave paintings weren't the only art form in the Upper Paleolithic. At Dolní Věstonice in Moravia, now part of the Czech Republic, archaeologists discovered a treasure trove of ceramic figurines. More than 700 statuettes have been recovered from this site, making it one of the richest assemblages of Paleolithic art known. And among these hundreds of figurines, woolly rhinoceroses are depicted again and again, showing the animal's unmistakable profile, captured in 3 dimensions in fired clay. Perhaps most remarkably, at Creswell Crags in England, archaeologists discovered what's known as the Pinhole Caveman, a late Paleolithic figure engraved on a woolly rhinoceros rib bone. This wasn't just an animal bone used as a canvas, it was meaningful that the image of a human was carved on the remains of a woolly rhinoceros, suggesting some special relationship or significance. The cave art of Europe provides us with something extraordinary, a direct window into how Paleolithic humans saw the animals around them. These weren't crude representations or symbolic images, they were careful observations rendered by skilled artists. When we look at these paintings, we're seeing woolly rhinoceroses, mammoths, bison, and horses through the eyes of people who encountered them every day, who lived with them, hunted them, and clearly held them in some special regard significant enough to spend hours painting them on cave walls deep underground. The oldest known cave art is even more ancient than Lascaux or Chauvet. In 2026, archaeologists announced the discovery of hand stencils on Muna Island in Indonesia, dated to at least 67,800 years ago. Even older are the geometric patterns at Maltravieso Cave in Spain, created by Neanderthals at least 64,000 years ago. And the oldest known narrative scene, a hunting scene from Sulawesi, Indonesia, dates to 51,200 years. But it's in Europe, during the Upper Paleolithic between roughly 40,000 and 10,000 years ago, that cave art reaches its peak of sophistication and frequency. The animals most commonly depicted are large herbivores, with horses, bison, aurochs, and deer dominating. But mammoths, woolly rhinoceroses, and other Ice Age megafauna appear regularly, testament to their importance in the lives and imaginations of Paleolithic peoples. The pigments used to create this art were carefully prepared. Red and yellow ochre, iron oxides, provided warm tones. Manganese oxide created blacks and dark browns. Charcoal from fires provided another source of black. These weren't paints in the modern sense, they were mineral powders mixed with water or animal fat, or sometimes just applied dry, blown onto the wall through hollow bones to create stencils and shaded effects. The artists had a sophisticated understanding of their materials and techniques. Why did Paleolithic people create this art? This question has occupied archaeologists and anthropologists for more than a century. Early theories suggested hunting magic, the idea that depicting an animal gave power over it, making hunts more successful. Others have proposed shamanic visions, with the deep caves serving as ritual spaces where altered states of consciousness produced visions that were then recorded on the walls. Some see the art as seasonal calendars, tracking the movements and behaviors of game animals. Still others interpret the images as narratives, stories being told and preserved for future generations. The truth is probably more complex than any single theory can capture. The motivations behind 30,000 years of cave art, created by thousands of different artists in hundreds of different locations, were likely varied and multifaceted. What we can say is that the animals depicted mattered to these people, enough to venture deep underground with primitive lamps, enough to spend hours carefully rendering their images, enough to return again and again to add new paintings to chambers already decorated. And what we see in these caves is not just artistic expression, it's also scientific observation. The accuracy of the animal depictions shows that these Paleolithic artists studied their subjects carefully. The proportions are correct, the behaviors are realistically rendered, the distinctive features of each species are captured with precision. When a Paleolithic artist painted a woolly rhinoceros with its characteristic fatty hump and long, curved front horn, they weren't imagining or stylizing, they were recording what they had observed. This accuracy has actually proved useful to modern scientists. When researchers want to understand what color patterns these extinct animals might have had, or how they held their heads when walking, or how their muscles created the distinctive body shapes, they can look to cave art as a kind of eyewitness testimony. The paintings provide information that no skeleton alone could tell us. The permafrost of Siberia, meanwhile, continued to yield its frozen treasures throughout the 20th century. Each discovery added pieces to the puzzle of Ice Age life, but some finds were more spectacular than others. In 1972, in the region called Chirapcha, between the Lena and Amur rivers in northeastern Siberia, local residents discovered something extraordinary protruding from a riverbank. It was a skull with ribs still attached, fur still clinging to the skin. As they carefully excavated around the find, they realized they had discovered a nearly complete woolly rhinoceros skeleton, with skin, fur, and even stomach contents preserved. The stomach contents were particularly valuable to scientists. By analyzing what the animal had eaten in its final meal, researchers could reconstruct not just what woolly rhinoceroses ate in general, but what was available to eat at that specific location and time. The stomach contained grasses and sedges, as expected, but also fragments of woody plants, including willow and larch twigs. This confirmed that woolly rhinoceroses had a more varied diet than simple grazing would suggest, and that the mammoth steppe ecosystem included both grasslands and scattered woody vegetation. 4 years later, in 1976, a different kind of discovery was made on the banks of the Aldan River, another Siberian waterway. A group of school children, exploring during their summer break, found bones protruding from the riverbank. When paleontologists arrived to investigate, they discovered a nearly complete woolly rhinoceros skeleton, and remarkably, both horns were still present. Finding the horns is rare because they are made of keratin, which normally decomposes quickly. But in the frozen conditions of permafrost, even this organic material could be preserved for 20,000 years. The Aldan River specimen's horns provided valuable data about horn growth and structure. Scientists could examine the growth rings in the keratin, similar to tree rings, which recorded the animal's life history. Periods of good nutrition showed up as thick growth bands, while periods of stress, perhaps harsh winters or times of food scarcity, appeared as thinner bands. The horns became a kind of diary, recording the animal's life year by year. The 21st century has seen even more remarkable discoveries emerge from the thawing permafrost. In 2007, erosion along the lower Kolyma River revealed a partial woolly rhinoceros carcass. The position of the remains suggested the animal had fallen into mud while alive, becoming trapped in an upward stretching position as it struggled to free itself. The preservation, while not complete, was sufficient for DNA extraction and analysis. The following year, 2008, was particularly productive. Near the Chukochia River, a nearly complete skeleton was discovered, adding to the growing collection of woolly rhinoceros remains that were allowing scientists to study population variation and genetics. And near the Amur River, additional mummified remains were found, these with particularly good soft tissue preservation. But perhaps the most significant frozen woolly rhinoceros discovery of the 21st century came in 2014. Two hunters, Alexander Banderov and Simeon Ivanov, were working near a tributary of the Semyulyk River in the Abaysky district of Yakutia when they found something remarkable protruding from a thawing riverbank. It was small, just a patch of brown fur at first, but as they carefully investigated, they realized they had found something extraordinary. It was a baby woolly rhinoceros, almost perfectly preserved. The calf was approximately 7 months old when it died, perhaps having fallen through thin ice or become separated from its mother and succumbed to the cold. The preservation was exceptional. The head, both horns, most of the fur, and even soft tissues were present and in good condition. The hunters, recognizing the scientific importance of their find, carefully transported the remains to the Yakutia Academy of Sciences. Scientists named the specimen Sasha after Alexander Bandarov, one of the finders. For the first time, researchers had access to well-preserved DNA from a young woolly rhinoceros, allowing them to study growth and development in ways that adult specimens couldn't provide. The DNA extracted from Sasha has proven invaluable for understanding woolly rhinoceros genetics and evolution. By comparing Sasha's genome to those of other woolly rhinoceros specimens from different time periods and locations, scientists can track how the population changed over time, how much genetic diversity existed, and how different regional populations related to each other. But Sasha wasn't the last remarkable frozen woolly rhinoceros to emerge from Siberia. In 2020, as rising temperatures caused by climate change melted permafrost that had been frozen for millennia, another extraordinary discovery was made. This specimen was older than Sasha, probably 3 to 4 years old at death, and the cause of death was clear: the animal had drowned, perhaps breaking through ice while crossing a frozen river or lake. What made this 2020 discovery particularly special was the exceptional preservation of internal organs. Most frozen specimens, even good ones, have experienced some decomposition of internal soft tissues. But this individual had apparently frozen so quickly after death that even delicate internal structures remained intact. Scientists could examine not just muscle and skin, but organs, providing an unprecedented window into woolly rhinoceros physiology. Additionally, this specimen preserved the small frontal horn, a feature that's usually lost even in otherwise well-preserved specimens. This allowed researchers to study how the two horns related to each other and how the smaller rear horn functioned in concert with the massive front horn. These frozen specimens from Siberia have revolutionized our understanding of woolly rhinoceros biology in ways that bones alone never could. We know what color their fur was, not from speculation, but from seeing the actual fur. We know how thick their skin was, how their internal organs were arranged, what they ate, how their horns grew, how their muscles attached to their bones. The permafrost has served as a deep freezer, preserving these animals in extraordinary detail for tens of thousands of years. But the frozen specimens also tell us something about climate and environment. The distribution of findings isn't random, they cluster in certain regions and time periods. This pattern reflects both where the animals lived and when and where conditions were right for preservation. The animals had to die in circumstances where they froze quickly, before scavengers could consume them and before decomposition progressed too far. Then they had to remain frozen continuously until modern discovery. This means that the specimens we find represent just a tiny fraction of the woolly rhinoceroses that actually lived. For every one that froze in just the right way and remained preserved, thousands or millions died and were consumed, decomposed, or were destroyed by geological processes. The frozen specimens are precious because they are so rare, improbable survivors from a vanished world. The same Siberian permafrost that preserved woolly rhinoceroses also preserved other Ice Age animals. Woolly mammoths are the most famous, with dozens of well-preserved specimens discovered over the past few centuries. But mummified remains of steppe bison, horses, cave lions, and even a woolly rhino calf's contemporary, a baby mammoth, have all emerged from the frozen ground. One of the most famous of all Ice Age mummies is a specimen called Blue Babe, not a mammoth or a rhinoceros, but a steppe bison. Blue Babe was discovered in July of 1979 near Fairbanks, Alaska, by a gold miner named Walter Roman. The bison had died approximately 36,000 years ago, killed by a cave lion, as evidenced by claw and tooth marks on the carcass. The nickname Blue Babe came from the bluish mineral coating that had formed on the mummy's skin, a mineral called vivianite that forms from the interaction of iron in the tissues with phosphates in the soil. Beneath this blue coating, the tissues were remarkably well preserved, including muscle tissue. In fact, the tissues were so well preserved that the research team at the University of Alaska Museum, after completing their scientific studies, did something extraordinary. They decided to see what 36,000-year-old bison tasted like. They carefully carved a portion of the neck muscle, stewed it with vegetables, and served it at a dinner party. According to participants, the meat was tough and somewhat mud-flavored, but recognizable as meat. It was, quite possibly, the oldest meal ever consumed by humans. Blue Babe, now on display at the University of Alaska Museum, stands as a testament to the quality of preservation possible in permafrost. The animal's pose, with head raised and mouth slightly open, captures a moment from 36,000 years ago, frozen in time. The steppe bison that Blue Babe represents, Bison priscus, was one of the most successful large herbivores of the Ice Age. These animals were considerably larger than modern American bison, standing over 2 meters tall at the shoulder and weighing more than 1,000 kilograms. Their horns were enormous, with a spread that could exceed 2 meters in the largest individuals. These were the bison that Paleolithic hunters pursued across the mammoth steppe, the bison depicted in cave paintings throughout Europe and Asia. Unlike the woolly rhinoceros, which went extinct roughly 14,000 years ago, steppe bison persisted into much more recent times. They survived the end of the Pleistocene and continued living into the Holocene, the current geological epoch. The last steppe bison lived in Alaska and died out around 3,450 years before present, making them one of the latest surviving members of the Ice Age megafauna. The extinction pattern of Ice Age megafauna wasn't uniform. Different species disappeared at different times and for different reasons. The woolly rhinoceros, highly specialized for very cold, dry conditions, and the mammoth steppe ecosystem, couldn't adapt when warming temperatures and increasing moisture changed its habitat. But steppe bison, more flexible in their diet and habitat preferences, survived through the dramatic climate changes at the end of the Pleistocene and persisted for thousands of years into a warmer world. Understanding these differential survival patterns is crucial for understanding the Pleistocene extinction event as a whole. It wasn't simply that the climate changed and all the megafauna died. It was more complex. Some species were more vulnerable than others, some regions lost their megafauna earlier than others, and multiple factors, climate, habitat change, human hunting, and disease, all potentially played roles. The frozen specimens from Siberia and Alaska provide some of the most direct evidence we have about the health and condition of Ice Age animals in their final millennia. By studying the bones, teeth, and tissues of these preserved individuals, scientists can look for signs of stress, malnutrition, disease, or population decline. Some late-period specimens do show signs of stress, suggesting that at least some populations were struggling even before they went extinct. But other specimens, even relatively late ones, show animals in good condition with no obvious signs of stress or decline. This suggests that extinction, when it came, may have been relatively rapid rather than a slow decline over many generations. Populations that seemed healthy could potentially be pushed over the edge by a single bad season, a disease outbreak, a pulse of human hunting pressure, or a sudden habitat change. The study of Ice Age extinctions is ongoing, with new evidence continuously refining our understanding. Each new frozen specimen, each new radiocarbon date, each new analysis of ancient DNA adds pieces to the puzzle. What's becoming clear is that there's no single simple explanation. The end of the Ice Age megafauna was the result of complex interactions between climate, ecology, and, in at least some cases, human activities. But beyond the scientific questions of how and why these animals went extinct, there's something deeply moving about the frozen specimens themselves. When you look at the photograph of Sasha, the baby woolly rhinoceros, or sea blue babe on display at the museum, you're not just seeing fossils or reconstructions. You're seeing the actual animals, their real fur, their actual skin, their preserved tissues. You're seeing them as they were in life more than 30,000 years ago. These specimens create a connection across the vast gulf of time. The people who painted woolly rhinoceroses on cave walls saw living animals that looked just like Sasha would have when fully grown. The hunters who pursued steppe bison across the mammoth steppe encountered animals just like Blue Babe. The Paleolithic peoples who carved spear throwers from woolly rhinoceros horn-handled material that came from animals just like the ones still emerging from the Siberian permafrost. And now, as climate change causes the permafrost to thaw at an accelerating rate, more specimens are being revealed. Each summer, more bones, more tusks, more mummified remains emerge from riverbanks and eroding cliffs across Siberia and Alaska. It's a race against time, because once these remains are exposed to warm temperatures, decomposition begins immediately. Specimens that survived for tens of thousands of years in the deep freeze can be destroyed in a single summer if they are not found and preserved quickly. Local residents and scientists have developed networks to report and recover these finds. When a mammoth tusk or a rhinoceros skull erodes out of a riverbank, there's often someone who recognizes its value and can alert authorities. Museums and research institutions maintain rapid response teams that can travel to remote sites to recover and preserve significant specimens before they are lost. This work is crucial not just for building museum collections, but for advancing scientific understanding. Each new specimen provides more data about population genetics, about diet and ecology, about climate and environment, about growth and development. The frozen specimens are time capsules, preserving not just individual animals but information about entire ecosystems. And what those ecosystems reveal is a world of stunning biological richness. The Ice Age wasn't a biological desert, it was a time of abundance. The mammoth steppe ecosystem supported some of the largest land mammals ever to exist, and it supported them in enormous numbers. The density of megafauna during glacial periods may have exceeded that of modern African savannas, which are themselves among the most productive ecosystems on Earth. This productivity was possible because of the unique character of the mammoth steppe environment. The cold, dry conditions limited decomposition, allowing nutrients to accumulate in the soil. The grasses and sedges that dominated the landscape were highly nutritious, similar to modern pasture grasses that are cultivated to support livestock. The long summer days in high latitudes meant extended growing seasons despite the cold. And the herbivores themselves played a role in maintaining the ecosystem, their grazing preventing the encroachment of woody plants and maintaining the productive grasslands. When the climate warmed and the mammoth steppe fragmented and disappeared, this entire ecosystem collapsed. The woolly rhinoceros, the mammoth, the steppe bison, the Irish elk, all of them were adapted for a world that no longer existed. The forests and wetlands that replaced the mammoth steppe couldn't support the same diversity or abundance of large herbivores, and without the herbivores, the specialized predators also disappeared. This transformation happened relatively quickly in geological terms, over just a few thousand years. But from the perspective of the animals living through it, the changes would have been almost imperceptible. Each generation would have experienced conditions just slightly different from the previous one. Gradually, the winters became wetter, the snowpack deeper. Gradually, shrubs and then trees began appearing in formerly open grasslands. Gradually, the ranges of familiar food plants shifted, contracted, and in some cases disappeared entirely. For highly mobile species, adaptation might have been possible. They could move, tracking the shifting climate zones and vegetation types that they depended on. And indeed, some species did manage this for a while. But for species that were highly specialized, that required very specific conditions to thrive, there was ultimately nowhere to go. When the mammoth steppe disappeared, so did they. The mammoth steppe wasn't just a Eurasian phenomenon. Similar grassland ecosystems existed in North America, where they supported their own assemblage of megafauna. And among the most impressive of these North American Ice Age animals was the Columbian mammoth. While woolly mammoths are the most famous members of the elephant family during the Ice Age, they weren't the only mammoths, and they weren't even the largest. That distinction belongs to Mammuthus columbi, the Columbian mammoth, which lived across much of North America from what is now Alaska down through Mexico, and possibly even into Central America. The Columbian mammoth was enormous, one of the largest land mammals to ever walk the earth. Males could reach heights of 3.72 to 4.2 meters at the shoulder, that's over 12 to nearly 14 feet tall. For comparison, a modern African elephant bull, the largest living land animal, stands about 3.3 meters tall. The Columbian mammoth was significantly larger. This size difference meant that Columbian mammoths weighed considerably more than any living elephant. Estimates for the largest males range from 8 to 10 metric tons, over 17,000 to 22,000 pounds. That's roughly twice the weight of a large African elephant bull. Some particularly large individuals may have exceeded even these impressive figures. The tusks of Columbian mammoths were similarly enormous. In large males, the tusks could exceed 4 meters in length and could weigh more than 100 kilograms each. These weren't straight tusks either, they curved dramatically, first growing outward and downward, then curving back inward and upward. In old males, the tips of the tusks sometimes even crossed in front of the trunk. The tusks served multiple purposes, as tools for stripping bark, digging for water or roots, and as weapons for combat with rival males and defense against predators. Unlike their woolly cousins in the north, Columbian mammoths didn't have thick coats of shaggy fur. They lived in more temperate climates, places where the summers were genuinely warm and heavy fur would be a liability rather than an advantage. They likely had sparse hair, similar to modern elephants, perhaps with slightly more coverage during winter months. This difference in fur covering is one of the key ways scientists distinguish woolly mammoth remains from Columbian mammoth remains in regions where their ranges overlapped. And those ranges did overlap, particularly in the northern parts of the Columbian mammoth's distribution. During the coldest periods of the Ice Age, the woolly mammoth's range extended well down into what is now the United States, while Columbian mammoths lived as far north as southern Canada. In these overlap zones, the two species encountered each other, and genetic evidence suggests they occasionally interbred, producing hybrid offspring that combined characteristics of both species. This interbreeding complicates the picture of mammoth evolution and extinction. Some individuals that have been classified as woolly mammoths based on their location or physical characteristics turn out to have significant Columbian mammoth ancestry when their DNA is analyzed, and vice versa. The genetic boundary between the two species was more porous than their physical differences might suggest. Columbian mammoths inhabited a variety of environments across their vast range. In the south, they lived in grasslands and parklands that were considerably warmer than the mammoth steppe of the north. They grazed on grasses, sedges, and browse from trees and shrubs. Analysis of their teeth shows wear patterns consistent with a diet that included significant amounts of grass, marking them as primarily grazers, though they certainly consumed other vegetation as well. These mammoths lived in social groups, almost certainly organized similarly to modern elephants. Females and their young lived in matriarchal herds led by the oldest and most experienced female. Males left these family groups when they reached adolescence and either lived solitary lives or formed loose bachelor herds. The large males would have been solitary much of the time, coming together with female herds only during breeding seasons. We know about Columbian mammoth social structure not just from analogy with modern elephants, but from fossil sites where multiple individuals are preserved together. At some sites, groups of females and young are found associated, suggesting they died together, perhaps trapped in mud, buried in a landslide, or killed in some other catastrophic event that claimed the entire family group. The relationship between Columbian mammoths and humans was significant. These were dangerous animals, certainly enormous and powerful, with formidable tusks and the intelligence of elephants. But they were also valuable prey. A single Columbian mammoth represented an enormous amount of meat, fat, hide, and ivory. 4 Paleo-Indian hunters, successfully killing a Columbian mammoth could feed a community for weeks. Archaeological evidence clearly shows that humans hunted Columbian mammoths. At multiple sites across North America, Columbian mammoth bones have been found in association with human tools and weapons. Stone projectile points are sometimes found embedded in mammoth bones or lying among mammoth remains in contexts that indicate human activity. The pattern of how bones were broken and distributed at some sites suggests deliberate butchery, with humans working to extract meat, fat, and marrow. One of the most famous Columbian mammoth sites is Blackwater Draw in New Mexico, where mammoth remains are associated with Clovis points, the distinctive stone projectile points used by some of the earliest widespread human cultures in North America. The Clovis people, who lived approximately 13,000 to 12,800 years ago, clearly hunted mammoths among other large game animals. But questions remain about the nature and extent of this hunting. Were mammoths a primary food source for Paleo-Indian peoples, hunted regularly and in large numbers? Or were they occasional targets of opportunity, with most mammoth hunting representing rare, perhaps desperate, events? Were humans taking down healthy adults, or were they primarily scavenging already dead animals and targeting sick, injured, or juvenile individuals? These questions matter because they relate to the contentious debate about what caused the extinction of Columbian mammoths and other North American megafauna. The timing is suggestive. The Columbian mammoth went extinct approximately 12,700 years ago, which coincides closely with both a major climate change event, the Younger Dryas Cold Period, and with the spread of human populations across North America. The overkill hypothesis, proposed by paleontologist Paul Martin in the 1960s, suggests that human hunting was the primary cause of North American megafauna extinctions. According to this theory, when humans first arrived in North America, they encountered naive prey animals that had no experience with human hunters and no appropriate fear responses. Human populations, facing abundant, easily killed prey, rapidly expanded and spread across the continent. But the prey populations couldn't sustain the hunting pressure, and species after species was driven to extinction. The hypothesis is controversial, and many scientists dispute it. Critics point out that climate change at the end of the Pleistocene was dramatic and certainly played a significant role in extinction. The replacement of open grasslands with forests, the disruption of established ecosystems, the changes in seasonal patterns, all of this would have stressed megafauna populations even without human hunting. More recent thinking suggests that the truth probably lies in the interaction between climate change and human activities. Climate change stressed megafauna populations and fragmented their habitats, making populations smaller and more isolated. Human hunting, which these reduced populations might have sustained under normal conditions, became unsustainable. Populations that were already struggling due to habitat loss and climate stress couldn't withstand even moderate hunting pressure. The combination of factors proved fatal. Columbian mammoth fossils have been found across their entire range, from Alaska to Mexico, but some sites are particularly rich. The tar pits at La Brea have yielded not just predators but also prey species, including Columbian mammoths. The Channel Islands off the coast of California have produced mammoth remains that tell their own unique story, which we'll explore shortly. And scattered across the Great Plains and the American West, mammoth bones and tusks emerge from riverbanks, eroded hillsides, and construction sites with remarkable frequency. One aspect of Columbian mammoth biology that has received particular attention is their growth rate and life history. Like modern elephants, mammoths were long-lived animals with slow reproductive rates. Females probably didn't reach breeding age until somewhere between 10 and 15 years old, similar to modern elephants. Gestation lasted approximately 22 months, again similar to elephants. And females probably gave birth to a single calf every 5 to 6 years. This slow reproductive rate meant that mammoth populations couldn't recover quickly from disturbances. If environmental conditions or human hunting caused a sudden drop in population, it would take many decades for numbers to rebound, assuming conditions improved. Species with fast reproductive rates, those that mature quickly and produce large numbers of offspring, can recover rapidly from population crashes. But species like mammoths, with their slow maturation and low birth rates, are vulnerable to extinction when populations decline. The last Columbian mammoths lived on the Great Plains and in the American Southwest approximately 12,700 years ago. By 12,000 years ago, they were gone. The continent that had hosted these magnificent animals for hundreds of thousands of years saw them disappear in a geological instant. But even as Columbian mammoths were vanishing from the mainland, a different story was playing out on the Channel Islands off the coast of California. There, a population of mammoths had been isolated for tens of thousands of years, and evolution had taken them in a dramatically different direction. The Channel Islands during the Ice Age were more accessible than they are today. Sea level was lower, sometimes more than 100 meters lower than present, because so much water was locked up in glacial ice. This meant that the distance between the mainland and the islands was reduced, and at times, several of the northern Channel Islands were connected to each other, forming a larger landmass called Santurce Island. Somehow, mammoths reached this island, most likely by swimming. Elephants are actually good swimmers and have been known to swim distances of several kilometers to reach islands. Once mammoths established a population on the Channel Islands, they were isolated from their mainland relatives. And in isolation, something remarkable happened. Island dwarfism is a well-documented evolutionary phenomenon. When large animals become isolated on islands where food resources are limited and there are few or no predators, natural selection favors smaller body size. Smaller animals need less food, can subsist on lower quality forage, and can maintain larger populations on limited island resources. Over many generations, the island population evolves to be significantly smaller than their mainland ancestors. This is exactly what happened to the Channel Island mammoths. Starting from Columbian mammoth ancestors that stood nearly 4 meters tall, the island population evolved down to just 1.72 to 2.02 meters tall at the shoulder, less than half the height of their ancestors. In weight, they were even more dramatically reduced, At perhaps 900 kilograms, they were only about 17% the size of a mainland Colombian mammoth male. These pygmy mammoths, classified as Mammuthus exilis, represent one of the most dramatic examples of island dwarfism known. In just a few tens of thousands of years, evolution produced a mammoth that would have stood only about chest-high to its mainland ancestors. The tusks were similarly reduced, short and relatively straight compared to the dramatically curving tusks of mainland Colombian mammoths. But despite their small size, these were still clearly mammoths. They retained the characteristic body shape, the domed heads, the columnar legs, all the distinctive mammoth features, just in miniature. From a distance, a pygmy mammoth might almost have looked like a baby Colombian mammoth. But these were fully adult animals, breeding and raising their own young on their isolated island home. The fossil record on the Channel Islands is extensive, with thousands of pygmy mammoth bones discovered over the years. Some of the best specimens come from Arlington Springs on Santa Rosa Island, where mammoth bones erode from the sea cliffs. The density of remains suggests that mammoths were abundant on the islands for a long period, successfully adapting to their limited environment. But the pygmy mammoths, like their larger cousins on the mainland, eventually went extinct. The youngest reliably dated pygmy mammoth remains are approximately 13,000 years old. This is slightly earlier than the final extinction of mainland Colombian mammoths, but within the same general timeframe of late Pleistocene extinctions. What caused the extinction of the pygmy mammoths is unclear. Climate change affected the islands just as it affected the mainland, though perhaps less dramatically. As sea levels rose with the melting of glacier, the islands became smaller and more isolated, with the formerly connected northern islands separating into Santa Cruz, Santa Rosa, San Miguel, and Anacapa. This reduction in available land area would have reduced the carrying capacity of the islands. There's also evidence that humans reached the Channel Islands by at least 13,000 years ago. The famous Arlington Springs man, a partial skeleton discovered on Santa Rosa Island, has been dated to approximately 13,000 years before present. If humans and pygmy mammoths coexisted on the islands, even a small amount of hunting pressure on a small island population could have been sufficient to drive extinction. The story of the pygmy mammoths is one of evolution and adaptation, showing how rapidly body size can evolve when selection pressures are strong. But it's also a story about vulnerability, a population isolated on a small island, with nowhere to retreat when conditions changed, was always at risk of extinction from any significant disturbance. Today, the Channel Islands National Park preserves the islands and their unique natural and cultural history. The fossil beds that contain pygmy mammoth remains are protected, and ongoing research continues to add to our understanding of these remarkable animals. The islands serve as a reminder that the Ice Age wasn't uniform. Different populations in different locations evolved in different directions, creating diversity within species that we're only now beginning to fully appreciate. But the story of Ice Age discoveries isn't just about finding fossils and frozen carcasses. In the modern era, the story increasingly involves DNA and the power of genetic analysis to reveal connections, relationships, and history in ways that previous generations of scientists could never have imagined. The idea of extracting DNA from ancient specimens was once considered impossible. DNA degrades over time, breaking down into smaller and smaller fragments until eventually nothing useful remains. In most fossils, the organic material has been entirely replaced by minerals, leaving no DNA at all. Even in frozen specimens where soft tissue is preserved, the DNA inside has been degrading for thousands of years. But in the 1980s and 90s, as techniques for working with very small amounts of DNA improved, scientists began to wonder if they could extract genetic material from Ice Age specimens. The first successful ancient DNA extraction from a fossil was published in 1984, from muscle tissue of a quagga, an extinct subspecies of zebra that had been preserved in a museum since the 19th century. This success opened the door to more ambitious projects. If DNA could be extracted from a museum specimen a century old, could it be extracted from Pleistocene megafauna tens of thousands of years old? The answer turned out to be yes, under the right conditions. Frozen specimens from permafrost, where cold temperatures had slowed decomposition and DNA degradation, were the best candidates. In 1994, scientists successfully extracted and sequenced DNA from a frozen baby mammoth. It was only a tiny fragment, but it was enough to begin reconstructing the mammoth genome. Over the following years, as technology improved, more and more mammoth DNA was sequenced. In 2008, a team published a nearly complete woolly mammoth mitochondrial genome, the genetic material contained in mitochondria, the energy-producing structures inside cells. But the real breakthrough came in 2015, when researchers published a complete nuclear genome sequence from a woolly mammoth specimen. This meant they had sequenced all of the genetic information encoded in the chromosomes in the cell nucleus, essentially the complete blueprint for making a mammoth. This was an extraordinary achievement, requiring the analysis of billions of tiny DNA fragments that had to be carefully sorted, aligned, and assembled like a jigsaw puzzle with millions of pieces. Having the complete mammoth genome opened up entirely new avenues of research. Scientists could now compare the mammoth genome to the genomes of living elephants, identifying exactly which genes had changed during the evolution of mammoths and their adaptation to cold climates. They could identify genes responsible for characteristic mammoth features like thick fur, small ears, and fat storage. One of the most interesting discoveries from the mammoth genome was the identification of genes related to cold adaptation. Mammoths had changes in genes that regulate body temperature, hair growth, and fat metabolism. They had modifications to hemoglobin, the protein that carries oxygen in blood, that allowed it to function efficiently at cold temperatures. These weren't random changes, they were specific adaptations that allowed mammoths to thrive in Ice Age conditions. Ancient DNA has also revealed relationships between different mammoth populations. By comparing genetic material from mammoths from different times and places, scientists can track how populations were connected, when they became isolated from each other, and how much genetic diversity they maintained. This has shown, for example, that woolly mammoth populations on Wrangel Island in the Arctic Ocean, which survived until approximately 4,000 years ago, were highly inbred and genetically depauperate, probably contributing to their eventual extinction. The analysis of ancient DNA has also clarified the relationships between different species. Woolly mammoths and Columbian mammoths, as we discussed earlier, turn out to have interbred where their ranges overlapped. The straight-tusked elephants of Europe, previously thought to be a separate species, show complex genetic relationships with both African and Asian elephants. The evolutionary history of the elephant family is more complicated than simple branching diagrams can capture. But ancient DNA isn't just about answering scientific questions, it has also opened up a controversial possibility: de-extinction. If you have the complete genome of an extinct species, can you bring that species back to life? In theory, yes. In practice, it's extraordinarily difficult and raises profound ethical questions. But that hasn't stopped people from trying. Several organizations and companies are working on mammoth de-extinction projects with various approaches and strategies. The most publicized effort is probably that of Colossal Biosciences, a company founded in 2021 with the explicit goal of bringing back the woolly mammoth. Their approach involves using CRISPR gene editing technology to modify the genome of Asian elephants, the closest living relatives of mammoths. By identifying the key genes that made mammoths different from Asian elephants and editing those genes into the elephant genome, they hope to create an elephant that has mammoth characteristics, or at least the most important ones. This wouldn't be a pure woolly mammoth, it would be a hybrid, an Asian elephant modified to have mammoth features like long fur, small ears, and cold adaptation genes. The project's supporters argue that this cold-adapted elephant could be reintroduced to Arctic regions where it might help maintain tundra grasslands, potentially even helping to slow permafrost melting by trampling snow and changing vegetation patterns. Critics raise numerous objections. Technically, it's unclear whether a creature produced by this process would actually be viable or healthy. Ethically, questions arise about creating animals that will live in captivity or in environments that may not be suitable for them. Ecologically, introducing genetically modified megafauna into modern ecosystems could have unpredictable consequences. And philosophically, many question whether resources devoted to de-extinction might be better spent on saving the many endangered species that still exist. The debate about de-extinction is unlikely to be resolved anytime soon, but the fact that we're having it at all is testament to how far the science of ancient DNA has come. From the first tiny fragments sequenced in the 1980s, we've progressed to complete genomes and serious discussions about bringing extinct species back to life. The frozen specimens that emerge from Siberian permafrost aren't just fossils anymore. They are potential genetic libraries, sources of information that could, perhaps, one day be used to resurrect vanished species. The technical challenges of de-extinction are formidable. Even with a complete genome sequence, creating a living animal requires much more than just genetic information. You need a viable cell with all its complex machinery, a surrogate mother to carry the pregnancy, and an environment where the resurrected animal can survive. For mammoths, the plan involves using Asian elephant eggs, editing their DNA to incorporate mammoth genes, and implanting the edited embryo into an Asian elephant surrogate. But which genes do you edit? A mammoth differs from an Asian elephant in potentially millions of small ways scattered across the genome. Some of these differences are critical, like the genes for cold adaptation and fur growth. Others might be less important, perhaps affecting subtle aspects of metabolism or behavior that we don't fully understand. Do you need to edit every difference to create a true mammoth, or would editing just the major adaptations be sufficient? And even if you successfully created a living creature with mammoth characteristics, what then? Where would it live? Modern Arctic tundra is not the mammoth steppe. The plant communities are different, the climate is different, and most importantly, the entire ecosystem that mammoths were part of no longer exists. A resurrected mammoth would be entering a world that has no place for it, a living anachronism, neither fully adapted to the modern environment nor able to recreate its vanished habitat. Proponents of mammoth de-extinction argue that cold-adapted elephants could actually help restore something like the mammoth steppe ecosystem. Their theory, called Pleistocene rewilding, suggests that large herbivores could transform modern tundra back into productive grassland by trampling shrubs, knocking down trees, and fertilizing the soil. Some even suggest this could help combat climate change by preventing permafrost thaw, as grasslands are thought to keep the ground colder than shrubland or forest. This idea has been tested on a small scale at Pleistocene Park in Siberia, where Russian scientist Sergei Zimov has introduced various large herbivores including horses, bison, and musk oxen to a fenced area of tundra. His observations suggest that the animals do indeed transform the landscape, trampling woody vegetation and promoting grass growth. But whether this could be scaled up, and whether it would actually be beneficial, remains controversial. Critics point out that the modern Arctic is fundamentally different from the Pleistocene. The climate is warmer, the seasonal patterns are different, and most crucially, the mammoth steppe ecosystem included many interdependent species, not just mammoths. You can't recreate a complex ecosystem by introducing just one species, no matter how ecologically important that species might have been. There are also questions about animal welfare. Is it ethical to create animals that will live their entire lives in captivity or in artificial, managed environments? Asian elephants are highly intelligent, social animals with complex emotional lives. A hybrid creature with elephant intelligence but mammoth characteristics would be neither elephant nor mammoth, it would be something new, created by humans for human purposes. What rights would such a creature have? What obligations would we have toward it? These ethical questions become even more complex when we consider that resources are finite. Every dollar spent on de-extinction is a dollar not spent on protecting endangered species that still exist. Asian elephants themselves are endangered, with populations declining due to habitat loss and human-elephant conflict. Is it more important to bring back a vanished species or to save a living one? The answer might seem obvious, yet the allure of de-extinction continues to attract significant funding and attention. Perhaps the most valuable contribution of de-extinction research isn't the possibility of bringing back extinct species, but rather the technologies being developed along the way. Advanced gene editing techniques, improved methods for working with elephant reproductive biology, better understanding of how genomes translate into physical characteristics, all of these have potential applications in conservation of living species. If we can figure out how to edit genes to make an elephant more cold tolerant, could we use similar techniques to help other endangered species adapt to changing climates? If we can work out the technical challenges of elephant reproduction in the laboratory, could that help breeding programs for the severely endangered Sumatran rhinoceros or Jovan rhinoceros? The development of these technologies also feeds back into paleontology and our understanding of extinct species. As we identify which genes are responsible for which characteristics, we can look at ancient genomes and make more informed inferences about what extinct animals were really like. We can move beyond simply noting that a woolly rhinoceros had different genes from a modern rhinoceros to understanding specifically what those genetic differences meant in terms of physiology, metabolism, behavior, and appearance. One particularly exciting application of ancient DNA technology is in understanding the process of extinction itself. By sampling multiple individuals from different time periods, scientists can track how genetic diversity changed as a species approached extinction. Did populations maintain healthy genetic variation right up to the end, suggesting rapid extinction from external causes? Or did genetic diversity decline over time, suggesting long-term population decline and inbreeding? Studies of the Wrangel Island mammoths have provided a particularly clear example. These mammoths survived on an isolated Arctic island until about 4,000 years ago, long after mainland populations had vanished. Ancient DNA from these late surviving mammoths shows severely reduced genetic diversity and accumulation of harmful mutations. The population was too small to maintain genetic health, and inbreeding was inevitable. While environmental factors likely delivered the final blow, the genetic evidence suggests this population was already in serious trouble. Similar studies of other late surviving populations can reveal whether genetic problems contributed to extinction or whether populations remained genetically healthy until external factors overwhelmed them. This isn't just academic curiosity, it has direct relevance to modern conservation. Many endangered species today exist in small, isolated populations. Understanding how Ice Age species fared in similar circumstances can help us predict and potentially prevent similar genetic deterioration in modern species. The woolly rhinoceros provides an interesting contrast to the Wrangell mammoths. Analysis of woolly rhinoceros genomes from different time periods shows that populations maintained substantial genetic diversity and large population sizes until very near the end. There's no evidence of a long, slow decline. Instead, the genetic data suggests that woolly rhinoceros populations were large and healthy until changing climate rapidly reduced their habitat. Extinction, when it came, was relatively quick. This pattern suggests that woolly rhinoceroses weren't slowly dying out over tens of thousands of years. They were successful, abundant animals right up until their habitat disappeared. This is actually a more frightening scenario in some ways than gradual decline, because it means that a species can seem perfectly healthy, with no warning signs of trouble, and still go extinct if environmental conditions change rapidly enough. For modern conservation, this is a sobering lesson. We can't assume that large populations and wide distributions guarantee a species' survival. If environmental change is rapid and severe enough, even abundant species can collapse quickly. The passenger pigeon provides a historical example. These birds literally numbered in the billions in the early 19th century, but a combination of habitat loss and hunting drove them to extinction by 1914. Large populations are no guarantee of survival if threats are severe enough. The study of Ice Age megafauna extinctions has also revealed the importance of what scientists call extinction cascades or secondary extinctions. When a keystone species goes extinct, it doesn't just disappear, it leaves a gap in the ecosystem that affects many other species. Predators that relied on that prey species must find alternatives or starve. Plants that depended on that animal for seed dispersal or pollination are affected. Prey species that benefited from the presence of that predator suddenly face different competitive pressures. When woolly mammoths went extinct, they weren't just one species disappearing. They were ecosystem engineers, animals whose activities shaped the environment for many other species. Mammoths knocked down trees, created trails, dug water holes, dispersed seeds, and fertilized vast areas with their dung. Their disappearance had ripple effects throughout the ecosystem. Some scientists have even suggested that the loss of mammoths and other large herbivores contributed to the expansion of forests and the change in Arctic ecosystems from productive grassland to less productive tundra. These ecosystem effects work both ways. Not only does the loss of one species affect others, but declines in prey species can doom predators. This is sometimes called second-order predation in the context of human impacts. When humans hunt large herbivores, they're not just affecting those prey species directly, they're also indirectly affecting the predators that depend on those prey. If human hunting reduces herbivore populations enough, predators face food scarcity and their populations decline as well. This phenomenon might explain some puzzling aspects of Ice Age extinctions. Why did predators like Smilodon and the short-faced bear go extinct at the same time as their prey, even though humans probably weren't hunting these predators intensively? The answer might be that direct hunting of predators wasn't necessary. Reducing their prey base was sufficient to doom them. Smilodon, so specialized for hunting very large prey, would have been particularly vulnerable to this effect. The short-faced bear, Arctodus simus, was one of the most remarkable predators of Ice Age North America. Standing over 3.5 meters tall on its hind legs, it was one of the largest bears that ever lived, larger than any modern bear species. Its long legs suggest it was a pursuit predator rather than an ambush predator, capable of running down prey over long distances. Some scientists believe it may have been primarily a scavenger, using its size to intimidate predators like saber-toothed cats and dire wolves away from their kills. Arctodus simus bones have been found across much of North America, from Alaska to Mexico, from coast to coast. These bears inhabited a variety of environments, from open grasslands to woodlands. They were clearly successful animals, yet they vanished at the end of the Pleistocene, extinct by about 11,000 years ago. What caused the extinction of the short-faced bear? Direct human hunting seems unlikely. These massive predators would have been extremely dangerous to hunt with Paleolithic weapons, and bear remains are relatively rare at archaeological sites. Climate change certainly affected them, as it affected all Ice Age megafauna. But many scientists believe that the key factor was the decline of large herbivores. If Arctidus relied on scavenging kills from other predators or on hunting large prey, the decline of megafauna would have reduced its food supply dramatically. The same pattern appears with other predators. The American lion, Panthera atrox, was another large predator that went extinct at the end of the Pleistocene. Larger than modern African lions, these cats probably hunted bison, horses, camels, and other large herbivores. As those prey species declined and went extinct, the American lion's food base disappeared, and the species followed its prey into extinction. The dire wolf, despite its name, was probably the least specialized of the large Pleistocene predators. It was a pack hunter, like modern wolves, and its robust teeth and jaws suggest it could eat a variety of prey and could crush bones to extract marrow. Yet even the dire wolf, with its flexibility and pack hunting strategy, went extinct, unable to adapt quickly enough when the megafauna it depended on disappeared. Understanding these extinction cascades is crucial for modern conservation. When we try to protect endangered species, we can't think about them in isolation. We need to consider the entire ecosystem, the web of relationships that connects predators and prey, herbivores and plants, pollinators and flowers. Protecting a single species might require protecting its prey base, its habitat, and perhaps even species that seem unrelated but play important roles in maintaining the ecosystem. The story of dire wolves has been revised recently thanks to ancient DNA studies. For many years, scientists classified dire wolves as Canis dirus, closely related to the gray wolf, Canis lupus. They were thought to be essentially just a larger, more robust version of living wolves. But DNA analysis revealed a surprise. Dire wolves were not that closely related to gray wolves at all. They diverged from the lineage leading to modern wolves, coyotes, and dogs about 5.5 million years ago, long before the genus Canis diversified into its modern species. This discovery was so significant that dire wolves were reclassified into their own genus, Enosine, meaning terrible wolf. Enosine dirus was not an oversized gray wolf but a distinct lineage that had been evolving separately for millions of years. This explains why dire wolves went extinct while gray wolves survived. They weren't just different species, they were fundamentally different kinds of canids with different evolutionary histories and adaptations. Gray wolves, Canis lupus, did survive the end Pleistocene extinctions, though their populations and ranges were severely reduced. Their flexibility, their ability to hunt prey from small to large, their social structure that allowed efficient exploitation of resources, all of these factors probably contributed to their survival. But they too faced massive changes as the megafauna disappeared. Wolves that had once hunted mammoths, bison, and horses had to adjust to a world with smaller, scarcer prey. The survival of some species and extinction of others provides crucial data about what makes species resilient. Generalists tended to survive better than specialists. Species with flexible diets did better than those dependent on specific prey. Animals with large geographic ranges and high population sizes had better survival odds than those with restricted ranges. And species with faster reproductive rates could recover from population crashes more readily than slow-breeding species. But there were exceptions to all of these patterns. Some generalists went extinct, some specialists survived. Distribution and abundance didn't guarantee survival. The passenger pigeon's fate makes that clear. The relationship between life history traits and extinction risk was probabilistic, not deterministic. Having characteristics associated with resilience improved your odds, but didn't guarantee survival. This complexity means that predicting which modern species are most at risk of extinction is not straightforward. We can identify risk factors, habitat specialization, small population size, restricted range, slow reproduction, but we can't predict with certainty which species will survive environmental change and which will not. This uncertainty makes conservation more challenging but also more urgent. If we wait until we're certain a species is doomed, we've waited too long. Conservation requires precaution, protecting species before they become critically endangered, preserving habitats before they are gone, acting on imperfect information because waiting for perfect information means waiting until it's too late. The fossil record provides countless examples of this principle. The woolly rhinoceros seemed perfectly fine, with large populations and wide distribution, until suddenly it wasn't. Columbian mammoths ranged across an entire continent, but went extinct in less than 1,000 years. Passenger pigeons numbered in the billions, but were gone within a human lifetime. Abundance today is no guarantee of survival tomorrow if conditions change rapidly enough. As we continue to study Ice Age extinctions, new analytical techniques reveal new insights. Stable isotope analysis, which looks at the ratios of different isotopes of carbon, nitrogen, and oxygen in bones and teeth, can reveal diet and habitat preferences with remarkable precision. By analyzing many individuals from different time periods, scientists can track how diets changed as extinctions approached, potentially revealing stress or shifting to lower quality foods. One fascinating application of isotope analysis at La Brea has revealed that Smilodon and dire wolves, despite living in the same place at the same time and both being large carnivores, had subtly different diets. Smilodon preferentially hunted animals from more forested environments, including forest bison and deer, while dire wolves tended toward prey from more open grasslands. This dietary separation would have reduced direct competition between these predators, allowing them to coexist. But as climate changed and forests expanded at the expense of grasslands, this balance would have been disrupted. The prey that Smilodon preferred would have become more abundant, while open country prey declined. On the surface, this might seem favorable for Smilodon, but ecosystems are complex. The expansion of forests brought other changes, perhaps new competitors, new diseases, or changes in seasonal patterns that affected prey distribution. And ultimately, the entire ecosystem collapsed, forests expanded further than the megafauna could tolerate, and both predator and prey disappeared. The study of extinction timing is also revealing new complexities. For many years, scientists thought that most Ice Age extinctions happened at roughly the same time, approximately 11,000 to 10,000 years ago, coinciding with the Younger Dryas cold period and the subsequent warming. But as more fossils are dated with increasing precision, a more complex picture emerges. Some species went extinct earlier than others. Woolly rhinoceroses disappeared from most of their range by 14,000 years ago, well before the Younger Dryas. Some populations of mammoths persisted much later, with Wrangel Island mammoths surviving until 4,000 years ago and some mainland populations lasting until around 10,000 years ago. Steppe bison in Alaska persisted until 3,450 years before present. Ground sloths in some parts of South America might have survived until as recently as 5,000 years ago, though this is debated. This temporal complexity suggests that there wasn't a single extinction event but rather a series of regional and species-specific extinctions playing out over thousands of years. Some species were more vulnerable to climate change, others were more affected by human hunting, and some faced a combination of factors that varied by region and time. The precision of modern radiocarbon dating has been crucial for revealing this complexity. Radiocarbon dating, remember, was developed by Willard Libby in the 1940s and earned him the Nobel Prize in Chemistry in 1960. The technique relies on measuring the decay of carbon-14, a radioactive isotope that organisms accumulate while alive but which begins decaying after death. By measuring how much carbon-14 remains in a sample, scientists can calculate how long ago the organism died. Early radiocarbon dating was relatively imprecise, with error ranges of several hundred years or more. But modern techniques, particularly accelerator mass spectrometry or AMS, have dramatically improved precision. AMS can date samples as small as 0.5 milligram of carbon, allowing scientists to date individual seeds, small pieces of bone, or single hairs. And the error ranges have been reduced to just a few decades in many cases. This precision has revealed that some species didn't decline gradually over millennia but rather disappeared rapidly from regions where they had been abundant. In some cases, layers of sediment show abundant remains of a species, then an abrupt absence in younger layers deposited just a few hundred years later. This pattern is more consistent with relatively rapid environmental change or sudden hunting pressure than with slow, gradual decline. The importance of accurate dating extends beyond just determining when species went extinct. It's crucial for understanding the relationship between human arrival and extinction events. In North America, there's extensive debate about exactly when humans first arrived. For many years, the Clovis culture, dated to around 13,000 years ago, was thought to represent the first human occupation. But discoveries of older sites have pushed back the date of human arrival to at least 15,000 years ago, and possibly earlier. If humans arrived in North America 15,000 years ago or more, but most extinctions didn't occur until 13,000 to 11,000 years ago, this suggests a gap of several thousand years between human arrival and extinction. This gap is difficult to reconcile with the simple overkill hypothesis, which predicts rapid extinction following human arrival. But it's consistent with more complex models where climate change and human impacts interacted over time to drive extinctions. In Australia, the timing is even more complex. Humans arrived in Australia at least 65,000 years ago, possibly as early as 70,000 years ago. But the timing of megafauna extinctions in Australia is hotly debated, with different studies providing dates ranging from about 45,000 years ago to as recent as 15,000 years ago for different species in different regions. The huge uncertainty in these dates makes it very difficult to determine what role humans played in Australian extinctions versus climate change and other factors. The challenge with dating in Australia is that most Australian megafauna fossils are found in caves or surface deposits that are difficult to date precisely. Radiocarbon dating only works reliably back to about 50,000 years ago, and many specimens are too old for this technique. Other dating methods, like uranium series dating or optical stimulated luminescence, can reach further back in time but have their own limitations and uncertainties. This is where the study of Ice Age extinctions becomes as much about developing better dating techniques as about understanding extinction itself. Scientists have to carefully evaluate each date, considering the dating method used, the material dated, the possibility of contamination, and the relationship between the dated material and the fossil of interest. A date on charcoal found near a bone tells you when the charcoal burned, which might or might not be the same time the animal died. These technical challenges mean that debates about extinction timing and causes continue, with new evidence regularly shifting scientific consensus. This is healthy for science. Certainty without evidence is dogma, but uncertainty with ongoing investigation is scientific progress. Each new fossil dated with improved techniques, each new analytical method applied to old specimens, each new site discovered and excavated, all of these contribute to refining our understanding. The radiocarbon revolution that Willard Libby initiated in the 1940s fundamentally changed archaeology and paleontology. Before radiocarbon dating, determining the age of fossils and artifacts was extremely difficult. Archaeologists could establish relative sequences, knowing that one layer was older than another, but couldn't assign absolute ages. Radiocarbon dating provided, for the first time, a way to establish timescales that transcended regional boundaries and connected findings from around the world into a unified chronology. The impact extended beyond just providing dates. Radiocarbon dating overturned many long-held theories about human prehistory. It revealed that many innovations, like agriculture or metallurgy, had been invented independently in different regions rather than spreading from a single origin. It showed that Stone Age peoples were capable of sophisticated achievements much earlier than previously thought. It demonstrated that the pace of cultural change varied enormously across different regions and time periods. In the context of Ice Age extinctions, radiocarbon dating provided the first solid evidence that extinctions occurred at different times in different places and affected different species at different rates. It revealed that some species survived much later than expected, like the Wrangel Island mammoths. It showed that extinctions in the Americas occurred around the same time as human arrival, strengthening arguments for human involvement. And it demonstrated that European extinctions began earlier and proceeded more gradually than American ones, suggesting different extinction dynamics in different regions. The development of calibration curves has further improved the accuracy of radiocarbon dating. It turns out that the ratio of carbon-14 to stable carbon-12 in the atmosphere hasn't been constant over time. It varies due to changes in cosmic ray intensity, which is affected by solar activity and Earth's magnetic field. It also varies due to changes in the carbon cycle, including how much carbon is stored in oceans, forests, and other reservoirs. To account for these variations, scientists have developed calibration curves based on tree ring sequences and other materials of known age. The INT-Cal calibration curves, most recently updated as INT-Cal 20 in 2020, allow radiocarbon dates to be corrected for atmospheric variation, significantly improving accuracy. A radiocarbon date of 10,000 years BP, corrected using calibration curves, might correspond to actual calendar years of 11,500 years ago, a difference of 1,500 years that could be crucial for understanding extinction chronology. These technical details might seem arcane, but they are fundamental to understanding Ice Age extinctions. Without accurate dates, we can't determine whether extinctions were rapid or gradual, whether they correlated with human arrival or climate change, or whether different species disappeared simultaneously or sequentially. Every refinement in dating techniques, every improvement in calibration curves, every new method for extracting datable material from difficult specimens, all of these contribute to a more accurate picture of what happened during this crucial period in Earth's history. But even as we look toward the speculative future of de-extinction, ancient DNA continues to illuminate the past. Every new genome sequenced adds to our understanding of Ice Age life. DNA from cave lions is revealing their relationship to modern lions and their own evolutionary adaptations. DNA from steppe bison is showing how they relate to modern bison and cattle. DNA from horses is revealing the complex history of equine evolution and domestication. Each extinct species whose genome is sequenced provides a new piece of the puzzle of life's history on Earth. The science of ancient DNA has also revealed something humbling. These extinct animals weren't that different from us or from the animals alive today. The mammoth genome is 99.4% identical to that of the Asian elephant. The genetic changes that separated a mammoth from an elephant, that created all of those distinctive adaptations for Ice Age life, amount to less than 1% of the genome. Small changes in DNA can produce dramatic differences in appearance and adaptation. This genetic similarity also means that the boundary between extinct and extant isn't absolute. If we can sequence the complete genome of an extinct animal, if we understand which genes create which characteristics, if we have a close living relative whose genome we can edit, then extinction becomes potentially reversible. This doesn't mean we should reverse it. The ethical and practical questions remain formidable. But the fact that it's technically possible changes how we think about extinction. At the same time, the study of ancient DNA reinforces the urgency of conservation. Every species that goes extinct represents the loss of millions of years of evolutionary history, a unique set of adaptations and characteristics that can never be exactly recreated. Even if de-extinction becomes routine, which seems unlikely, it would only be possible for species that left well-preserved genetic material behind. Most extinctions leave no such material. Once a species is gone, it's gone forever. The frozen ground of Alaska and Siberia continues to yield new specimens, new data, new DNA. But time is running out in a sense. Climate change is thawing permafrost at an accelerating rate, and once specimens are exposed and thaw, degradation proceeds rapidly. There may be thousands or tens of thousands of frozen Ice Age animals still entombed in permafrost, waiting to be discovered. But each year, some are lost as the ice melts and the organic material decomposes. It's a race to find and preserve these specimens before they are gone. Meanwhile, in labs around the world, scientists are still decoding the information locked in specimens that have already been recovered. Each frozen mammoth, each preserved rhinoceros, each mummified bison contains stories in its DNA, in its tissues, in its bones. These stories tell us about adaptation and evolution, about ecosystems and climate, about survival and extinction. They are stories from a vanished world, but they have lessons for our world today. As we face our own period of rapid environmental change, the story of Ice Age extinctions becomes more than just a historical curiosity. It becomes a case study in how megafauna respond to changing climates, changing ecosystems, and human presence. The Pleistocene extinction wasn't inevitable. Different species responded to similar challenges in different ways. Some, like the steppe bison, survived for thousands of years into the Holocene. Others, like the woolly rhinoceros, disappeared quickly. Understanding what made the difference could help us protect modern megafauna facing their own extinction crises. The more we learn about Ice Age animals, the more we realize how much we still don't know. Every answer generates new questions. The frozen specimens that seemed like the final word on what these animals looked like turn out to raise questions about color variation, seasonal changes, and individual differences. The DNA that promised to reveal all of mammoth biology shows us the genes, but we still have to figure out exactly how those genes translated into living, breathing animals. The fossil sites that seemed fully excavated continue to yield new specimens and new insights. This is the nature of paleontology, the science of ancient life. It's detective work, piecing together evidence from fragments and traces to reconstruct vanished worlds. Every new discovery can confirm previous hypotheses or overturn them entirely. The field is dynamic, constantly evolving as new technologies become available and new specimens are found. Consider what we now know about saber-toothed cats compared to what we knew a century ago. When the La Brea excavations began, Smilodon was known primarily from scattered bones. Scientists could reconstruct the skeleton, could see the remarkable canine teeth, but almost everything else was speculation. How did it hunt? Was it solitary or social? How fast did it grow? What exactly did it eat? Now, thanks to the wealth of specimens from La Brea and advances in analytical techniques, we can answer all of these questions with reasonable confidence. We know Smilodon was an ambush predator that used its powerful forelimbs to wrestle prey. We have strong evidence it was social. We understand its growth rate and development. We can identify its prey through isotope analysis. We can even diagnose specific injuries and diseases in individual animals from thousands of years ago. This level of detailed understanding, moving from bare bones to behavioral ecology, represents one of the great achievements of 20th and 21st century paleontology. And it's been accomplished not through any single breakthrough, but through the steady accumulation of evidence, the development of new analytical methods, and the careful, systematic study of thousands of specimens by hundreds of researchers over many decades. The same process is playing out with other Ice Age species. Our understanding of woolly mammoths has been transformed by frozen specimens and ancient DNA. Our picture of woolly rhinoceroses has been filled in by mummified remains from Siberia. Our knowledge of Ice Age ecology has been revolutionized by detailed studies of sites like La Brea that preserve entire communities of interacting species. But for all that we've learned, mysteries remain. Why did some species survive the end Pleistocene extinction while others perished? What role did disease play, if any? How did climate change interact with human hunting to drive extinctions? What were the secondary ecological effects as key species disappeared? How did surviving species adjust to the loss of so many other members of their communities? These questions keep paleontologists busy, and they'll likely keep them busy for a long time to come. Every fossil site is unique, every specimen adds data, every new analytical technique opens new lines of investigation. The study of Ice Age animals is far from complete, and new discoveries continue. In just the last few years, mammoth remains have been found in unexpected places, cave paintings have been discovered on islands where they were previously unknown, new species have been identified from fossil material that had sat in museum collections for decades, new dating techniques have refined our understanding of when extinctions occurred, and ancient DNA studies have revealed previously unknown population structure and gene flow. One of the most exciting recent developments is the use of environmental DNA, or EDNA. This technique doesn't require finding actual remains of animals. Instead, it looks for traces of DNA that organisms leave in the environment. Animals shed skin cells, urinate, defecate, and bleed, all of which deposit DNA into soil and sediment. In permafrost and other well-preserved sediments, this DNA can persist for thousands of years. By carefully extracting and sequencing EDNA from dated sediment layers, scientists can identify which species were present at a location and time even if no fossils of those species have been found. This technique has the potential to revolutionize our understanding of past ecosystems, filling in gaps in the fossil record and detecting rare species that left few physical remains. However, EDNA studies also come with challenges. DNA can move through soil, potentially contaminating older or younger layers. The absence of DNA doesn't necessarily mean a species wasn't present, it might just mean conditions weren't right for preservation. And identifying species from tiny fragments of degraded DNA can be difficult, requiring very careful analysis to avoid errors. Despite these challenges, EDNA is already producing interesting results. Studies of sediment cores from the Arctic have extended the apparent survival dates of some species and provided evidence for the presence of species in regions where they weren't previously known. As techniques improve, EDNA will likely become a standard tool in paleontological research. Another frontier in Ice Age paleontology is the application of advanced imaging and computational techniques. CT scanning of fossils can reveal internal structures without damaging specimens. Three-dimensional modeling allows researchers to test hypotheses about biomechanics, to simulate how joints moved or how teeth functioned. Stable isotope analysis of teeth can reveal dietary information and even track seasonal migrations. Trace element analysis can provide information about the geology and environment where an animal lived. These techniques generate enormous amounts of data, and making sense of that data requires computational power and sophisticated statistical analysis. Modern paleontology is increasingly a data science, with researchers spending as much time at computers as they do in the field or laboratory. This technological sophistication has allowed researchers to extract more information from specimens than ever before. A single mammoth tooth can now yield data about the animal's age at death, its diet, where it lived, what season it died in, whether it was healthy or stressed, and its genetic relationships to other mammoths. Each specimen becomes a rich source of information about individual life histories and broader population patterns. As our understanding of Ice Age animals deepens, we're also gaining appreciation for just how rich and complex Ice Age ecosystems were. The mammoth steppe supported an assemblage of large mammals that has no modern equivalent. The diversity and abundance of megafauna exceeded anything we see today, even in the most productive modern ecosystems. Why was the Ice Age so favorable for large mammals? Part of the answer lies in the characteristics of the mammoth steppe ecosystem. The cold, dry climate limited decomposition, allowing nutrients to cycle efficiently. The grasses that dominated the ecosystem were highly nutritious, supporting large populations of herbivores. The herbivores themselves helped maintain the grasslands through their grazing, creating a feedback loop that sustained the system. Another part of the answer may lie in the long duration of Pleistocene glacial cycles. Large mammals, with their slow reproductive rates, need stable conditions to evolve and diversify. The Pleistocene provided that stability, with glacial periods lasting tens of thousands of years, long enough for megafauna to become highly adapted to Ice Age conditions. When that stability was disrupted, when the climate shifted from glacial to interglacial conditions and the shift happened relatively rapidly, the finely tuned adaptations of Ice Age megafauna became liabilities. Woolly rhinoceroses, so perfectly adapted for cold, dry grasslands, couldn't survive in warmer, wetter forests. Columbian mammoths, requiring vast areas of productive grazing, couldn't maintain populations when habitats fragmented. Smilodon, specialized for hunting very large prey, faced starvation when its prey declined. The lesson, perhaps, is that specialization is a double-edged sword. In stable conditions, being highly specialized for your particular ecological niche is an advantage. You can outcompete less specialized rivals, you can exploit resources more efficiently. But when conditions change, that specialization becomes a trap. The more finely adapted you are to one set of conditions, the harder it is to adapt when those conditions change. This is relevant today as we face rapid climate change driven by human activities. Modern species are adapted to current conditions, and rapid change will disrupt those adaptations just as it disrupted Ice Age megafauna. The species most at risk are likely to be specialists, those with narrow habitat requirements, specific food needs, or limited ranges. Generalists, species that can eat a variety of foods, tolerate a range of conditions, and adjust to changing circumstances, are more likely to survive. But even generalists have their limits. If change is rapid enough or extreme enough, even flexible species may not be able to adapt quickly enough. And for large mammals with slow reproductive rates, the challenge is even greater. They can't evolve rapidly, they can't produce enough offspring to explore a wide range of genetic variations, they need time to adapt. Time that rapid environmental change doesn't provide. The parallels between the late Pleistocene extinction and our current biodiversity crisis are sobering. Many of the same factors that drove Ice Age extinctions are at play today: rapid climate change, habitat loss and fragmentation, human hunting and persecution, and the interaction between all of these stresses. Modern elephants, rhinoceroses, big cats, all the living relatives of Ice Age megafauna, face similar challenges to those that overwhelmed their Pleistocene cousins. But there's also a crucial difference. The humans who lived at the end of the Pleistocene didn't understand what was happening. They couldn't have known that their hunting was contributing to extinctions or that the climate was changing in ways that would reshape ecosystems. They lacked the knowledge and the tools to prevent extinctions even if they had wanted to. We don't have that excuse. We understand what's happening. We know that species are going extinct at rates that far exceed background extinction rates. We can see the causes, and we have the capability to act. Conservation efforts can work. Protected areas can preserve critical habitats. Regulated hunting can prevent overexploitation. Captive breeding programs can rescue species from the brink of extinction. Whether we choose to act with sufficient urgency and commitment remains to be seen. The story of Ice Age animals and their discoveries is ultimately a story about time, deep time, the vast spans of geological history during which species evolve, thrive, and go extinct. The woolly rhinoceros existed as a distinct species for over 400,000 years. That's roughly 2,000 times longer than recorded human history. From the woolly rhinoceros's perspective, the entire span of human civilization, from the first cities to the present day, would be barely a moment. Yet that moment has had outsized significance. In just a few thousand years, humans have transformed from just another species of large mammal to a force that shapes the entire biosphere. We've driven countless species to extinction, altered the climate, reshaped landscapes, and changed the chemistry of the atmosphere and oceans. We've become a geological force, creating what some scientists call the Anthropocene, a new epoch defined by human impact on Earth systems. The discoveries of Ice Age animals, the careful excavation of fossils, the analysis of frozen specimens, the sequencing of ancient DNA, all of this scientific work serves a purpose beyond satisfying curiosity. It provides perspective. It shows us that the world has not always been as it is now, that climates change, that ecosystems transform, that the species composition of biological communities is not fixed and permanent. But it also shows us that extinction is real and permanent. The woolly rhinoceros is not coming back, regardless of what de-extinction enthusiasts might hope. The Columbian mammoth is gone, the Smilodon is gone, the ground sloths and glyptodonts and Irish elk are gone. They existed for hundreds of thousands or even millions of years. They were successful beyond any measure that matters to evolution, and now they are gone. Their absence has left a world that is less diverse, less dramatic, diminished in ways we can measure and in ways we probably don't fully understand. As you drift off to sleep tonight, think about those mammoths emerging from the permafrost, perfectly preserved, their fur still thick and brown after 30,000 years. Think about Marcel Revivat, just 18 years old, descending into Lascaux's cave with a makeshift lamp and discovering walls covered in ancient art. Think about the researchers at La Brea carefully excavating another Smilodon skull from the tar, adding one more specimen to a collection that numbers in the hundreds of thousands. These discoveries connect us to deep time, to a world so different from ours that it might as well be another planet. Yet it was this planet, just a few thousand years ago. The animals were real, the ecosystems functioned, life thrived in abundance. And then it changed, rapidly, drastically, and most of the megafauna didn't survive the transition. We live in the aftermath of that transition, in a world diminished by those extinctions. But we also live in a time when we can still make choices about what survives and what disappears. The elephants and rhinoceroses and big cats that remain are our connection to that vanished world of giants. Whether they survive depends, in large part, on us. So as you rest tonight, remember the woolly rhinoceros with its long horn sweeping snow aside to reach the grasses beneath. Remember the Smilodon cubs waiting at the den while their mother hunted. Remember the Columbian mammoths migrating across grasslands that stretched to the horizon. Remember that they were here, that they mattered, and that their story, preserved in frozen flesh and fossil bone and cave paintings, is part of our story too. If you enjoyed this journey through time, learning about the discovery of Ice Age animals, please consider liking this video and subscribing to the channel. Your support helps us create more content exploring the wonders of the natural world and the deep history of life on Earth. Rest well, and perhaps you'll dream of mammoths.